Hello and welcome back. Today we are going to finish off Unit 2 with our final set of notes. This is sort of like an introductory set of notes on photosynthesis. This is a brief overview. This will be discussed in much more detail when we get to topic 8. But let's get started anyways, because I think photosynthesis is interesting. So, what is it? It is just a process where organic compounds are synthesized from inorganic molecules in sunlight. And what that means is basically that plants take inorganic molecules, and inorganic just means non-living, basically, molecules, and they can synthesize an organic compound. In this case, that organic compound is glucose. This all happens in the chlorophyll, in the chloroplast, which are, again, found in plants. I think you guys have a general idea of what photosynthesis is. And, of course, photosynthesis uses light energy, right, light from the sun, to create chemical energy, which is ATP. And ATP is sort of the energy that is used for nearly all processes that happen in living organisms. Now, we care about the light spectrum, of course we do, but we care specifically about the sun and where it emits its peak power. So this happens in the visible region of the light spectrum, and in this case that's from 400 to 700 nanometers, and that's this area down here. Now there are other parts of the light spectrum that we are not going to talk about, like UV light, X-rays, right, gamma rays. These are not visible wavelengths for us as humans, and so yes, they exist, but we are going to focus in on the visible spectrum, which again is 400 to 700 nanometers. So the colors within the visible spectrum are different wavelengths, and you can see that because you have these kind of larger wavelengths where there's like more space in between them, versus these like tight wavelengths where there's not a lot of space in between them. Okay, so longest wavelengths to shortest wavelength. Now, I mentioned the chloroplast earlier, but this is where the magic happens, okay? This is the organelle in plants that is responsible for photosynthesis. So photosynthesis happens within the chloroplast, just like cellular respiration happens within the mitochondria. In the chloroplast, there are membrane sacs called thylakoids, and thylakoids are these little things right here that look like stacks of coins, and when they're arranged into their stacks, they're called grana. So thylakoids are the singular coin, but when you have a stack of coins, they're called grana. They contain chlorophyll, which is the pigment, and it's the site of the light-dependent reactions. So we'll get into light-dependent and light-independent reactions in a moment, but that's like, again, that's like really important to know the location of this. And then the fluid surrounding all of this is called stroma. So in this picture, it's this blue stuff. That's called stroma, and it contains carbon-fixing enzymes. That's where light-independent reactions happen. Okay, so dependent reactions happen within the thylakoids, and the independent reactions happen in the surrounding space. So, continuing on with our kind of deep dive into the chloroplast and chlorophyll, I had mentioned that chlorophyll is a pigment. It is a pigment. It's a green pigment. It's what makes leaves green, found in photosynthetic organisms, and it's responsible for light absorption. So chlorophyll is where the light gets captured. So as it absorbs the light, it releases electrons, which are then used to synthesize ATP. So basically, once light is absorbed, electrons go out into the world of the chloroplast, find ATP, and say, let's go, buddy, like, things are happening. Okay? Different chlorophyll molecules can do different things, okay? They absorb light most strongly in the blue portion of visible light, followed by red. So blue light will make plants grow the best, and it reflects light most strongly in the green portion. That's why leaves are green. Because green reflects light. And so when light hits the green plants, it will reflect off of it, but it will also get absorbed in the chlorophyll because it's a green pigment, so it will take in that light. So if you were to ever grow plants in different colored lights, green light is actually the worst to grow in. And you might have thought that it would be the best because plants are green, green is good, green is very environmental, but no, the opposite. Blue is the best. Now, photosynthetic organisms don't rely on just this one single pigment to absorb light. Okay, they're grouped into what's called a photosystem, and those photosystems absorb light energy. When you group pigments together, you can maximize light absorption, right? Working as a team is always easier than working alone. So we're focusing in on this one singular pigment, chlorophyll, but there's actually a number of pigments and a number of photosystems that do this type of work. And these accessory pigments also help explain why all leaves are not green, right? Chlorophyll is green, but there are other pigments like anthocyanins, which are different colors. There are pigments that are yellow, that are red, that are different shades of green, that are orange, right? It's just they produce different colors. And deciduous trees change color when leaves stop producing chlorophyll in the winter. Now, I know you've seen this, right? You've seen when fall is starting and you're getting closer to winter, and all of a sudden the leaves on the trees turn yellow and red and orange, and it's beautiful and it's great, but it actually just means that the tree itself isn't producing chlorophyll anymore. So the pigment that used to color the leaves is just not doing its job anymore. And so these accessory pigments are there to pick up the slack, but it changes the colors of the leaves. Now, action spectrum and absorption spectrum. So this is talking about where light can be absorbed by each pigment and then the overall rate of photosynthesis. So when we're talking about the absorption spectrum, I had mentioned that blue is the best, and you can see that right here, that we have the best absorption happening. Our highest peaks are right here. We have nearly nothing happening in green and yellow, and then our second highest peak is in this sort of reddish area. So blue is our winner, and then red is like the second place, but still silver medal, and then green, like, sorry dude, you're not doing great. But you can also see that you have different pigments that we're looking at, right? So carotenoid, we have chlorophyll A, chlorophyll B. These are different colored pigments. So our accessory pigments that we had previously talked about. And then the action spectrum, this is talking about photosynthesis. So if I can absorb more light, I can do more photosynthesis. So you see the peaks are sort of mirrored in what the expectation, like the expectations are what you would assume them to be. So we have our highest peak again in this sort of blue, purple sort of, but mostly blue range, right? Right here we have our highest peak. However, our red peak is significantly higher in the action spectrum than it is in the absorption spectrum. Okay, so although we're absorbing less light, we can still have a high rate of photosynthesis because we're still absorbing a decent amount of light. Between chlorophyll A and B, we're absorbing quite a bit of light. So I had mentioned that there are two steps. Well, I mentioned that there was light dependent and light independent. You may have been able to figure out what that means at this point. Light dependent means that you need light to do the reaction, right? It's dependent on light. Whereas light independent is you don't. The reaction will occur whether light is there or not. So in the first step, light dependent, we have the light being absorbed by the chlorophyll which results in the production of ATP. That we've already talked about. Light is also absorbed by water, which then splits the water molecule into its main components, which are oxygen and hydrogen. And then hydrogen and ATP are used in the step 2 reaction, the light independent reaction, and oxygen is released as a waste product. We know that in photosynthesis, you take in CO2, you take in water, and you take in light, and then the waste products are glucose and oxygen, and this is where one of the waste products is formed. Oxygen is released as a waste product in the light-dependent reaction. And then when we get to step 2, right, we have the ATP and the hydrogen from step 1, and then hydrogen and the carbon dioxide are combined, and they form organic compounds like carbohydrates and amino acids. The reason that we need ATP there is because we need the energy. We need that energy to be in the independent reaction to power all of these reactions to form these organic compounds. When the ATP is there, it fixes the carbon molecules together to be able to build up these polymers like carbohydrates, and amino acids will be formed into proteins. So all of this is necessary to do all of these sort of like behind the scenes reactions that plants need just to survive, but it is broken down into 2 very distinct steps. So again, light energy comes in, it's absorbed by both water and chlorophyll. Water is broken down, oxygen is a waste product, hydrogen moves on to the next step, and chemical energy is ATP. So that is produced and it continues on. And then carbon dioxide is brought in just as like part of photosynthesis. The formula calls for you need carbon dioxide to do photosynthesis. And so when carbon dioxide comes in, it is put together with all of the final pieces of your light-dependent reaction, right? NADPH and ATP. You go through those different reactions and you are creating organic compounds. So, again, you start with light, you end with organic compounds like carbs and proteins. There are some limiting factors, and we've talked about limiting factors on enzymes before. They are similar limiting factors on photosynthesis. So temperature, again, enzymes can be natured if temperature is too high, and they also slow down significantly if temperature is too low. Light intensity, chlorophyll are photoactivated, but will eventually plateau as the chlorophyll becomes saturated. We've seen this before when we're talking about substrate concentration. So this is the same thing, right? We are going to plateau eventually. Same thing with carbon dioxide. As you add carbon dioxide, it will increase pretty dramatically, but it will eventually plateau because, again, you are saturating all of the enzymes and all of the things that are responsible for absorbing the light and absorbing the carbon dioxide. There's only so many that are available, and once they're being used, you can't just like magically make more. So you have to wait until they go through their steps and then they release the CO2, or they're ready for more light, whatever it may be, and the process will continue on, but the rate of reaction will not increase. Okay, let's talk about chromatography. This is an experimental technique, and this is a way to separate pigments so that you can see what pigments you're actually working with. So you make a mixture and you dissolve it in a specific fluid, and then you pass it through a specific material. I know. We are going to do this in class, so you will see how this actually works, but the key here is that components travel at different speeds. They travel at different speeds because they are different weights. So larger pigments travel slower because they're bigger and they weigh more, whereas smaller components will travel faster and they will travel higher on the chromatography paper. because of these different weights, they separate out from each other. So you can start with one solid color, and then you can end up breaking up that color into its base components, so you can see what pigments are actually in there. And then from that, you can calculate an RF value. And an RF value is literally just how far the component traveled over how far the solvent traveled. So how far did the pigment itself travel versus how far did like the chromatography liquid travel overall. And there are two types of chromatography. We are going to do paper chromatography in class. It's cheaper, but it works just the same. So there's paper chromatography, which uses paper. That's the static material that the fluid is passed through. Or you can do thin layer chromatography, which uses like silica gel. It runs faster, it has better separation. It also is significantly more expensive, so we're not using that one. But it's very cool and it's very visual, so you will be able to see very quickly what's happening. So this is an example of the paper one that we're going to do in class. So you have your liquid at the bottom, you have your chromatography paper here, and you are going to let the water run up. And you can see that the pigments will start to separate. You have this sort of like, this is your like original stain here. So this is where all of the pigments will start. And then as the water runs through, the chromatography liquid runs through, the pigments will separate out based on weight. And you can see that they all will be in their own area. It might not be like this clear, but you will be able to very distinctly see like different color pigments throughout the chromatography paper by the time you're done. If you're not careful, the pigment will run right off the paper, so you do have to keep an eye on it. Eventually, if you blink too long, or take a little nap or something, the orange will just fly right off the paper, and then you think, oh no, like I didn't have any carotenes in this leaf stain, when in reality you did. You just you missed it. It's gone now. And then you can calculate the RF value, again, based on how high the solvent went versus the pigment. You can see that this pigment is very close to the top of the solvent, so that is why it's very close to 1, whereas chlorophyll B is at the bottom, so it's very far away, so it has a much lower RF value. And then one of the last things we're going to talk about is the oxygenation of the Earth. So approximately 2.3 billion years ago, that's what BYA is, billion years ago, photosynthetic organisms began to saturate the environment with oxygen. So prior to that moment in time, the atmosphere did not have a lot of oxygen in it. Even now, oxygen only makes up about 21% of the atmosphere, so yes, it's definitely there, but it's not as high as maybe you would think it is. But anyway, so what happened when oxygen started to saturate the environment? Well, there was some there was some impact on oceans, atmosphere, rock deposition, and biological life. So in the oceans, originally you had high levels of iron, and when oxygen reacts with iron, you form iron oxide, okay, which is not ideal. When iron was completely consumed, oxygen gas accumulated in the atmosphere. So it started off in the oceans where it kind of met with iron and formed this product, iron oxide. Eventually, it just was too much oxygen, and so it went into the atmosphere. The first 2 billion years of the Earth, the atmosphere was anoxic, which means that there was no oxygen in there, which is what I was saying earlier. But now it makes up about 20% of the atmosphere. Again, I jumped the gun on that one. I had already said that, but it is also right here. So then, continuing on with what this sort of photosynthetic organism outcome is the reactions in the oceans, right, this formation of iron oxide caused deposits called banded iron formations. When these iron formations formed, the deposition slowed in oceans and these iron-rich layers started to form on land. So that's good. We like iron on land, but we don't want to be overrun with iron. And from that, biological life was sort of able to evolve. When you have free oxygen molecules, just like out in the world, it's toxic to anaerobes, right? They do not survive with oxygen. Anaerobic means without oxygen. So this free oxygen ended up killing off a lot of anaerobes, which is interesting because for you as a human, billions of years later, it's critical, right? You need it. You need it to survive. So once these anaerobes were wiped out, unfortunately, many were wiped out unless they were in the proper habitat to be able to make quick changes and find a way to survive. Yeah, they pretty much died. But the aerobically respiring organisms like plants, like animals, like humans were able to then evolve in this new world. So again, this is just sort of like showing you a timeline. In this area, we have no oxygen in the atmosphere. In this area, we are starting to have an increase in oxygen, but it's mostly being absorbed by oceans, and you're forming that iron oxide. Eventually, you start to get too much oxygen formation in the oceans and starting to bring itself into the atmosphere. And then you see this really take off. And this is when the oxygen sinks are full in the ocean and the atmosphere is really just accumulating all that excess oxygen. And this is where, you know, you start to see the decrease of anaerobic organisms and the beginning of aerobically respiring organisms, the beginning of their lives. Okay, so that is it for Unit 2. You did it, you made it. Proud of you, and I'll see you in class.